Excellencies, distinguished guests, respected faculty members, dignitaries from industries and academia, and beloved students of Daffodil International University. Assalamu alaikum. We are about to start today's seminar on cancer genetics and precision medicine organized by Department of Pharmacy and Department of Public Health, Daffodil International University.
I welcome you all to the prestigious scientific seminar to be started soon. I am so pleased to be a part of the program organized by the Department of Pharmacy in collaboration with Department of Public Health, Daffodil International University. We are going to learn and discuss about a disease which is the second leading cause of death worldwide. Bangladesh currently has more than 1.5 million cancer patients, according to a recent WHO report. Alarmingly, this number is increasing day by day. Today, when anyone is diagnosed with cancer, he or she usually receives the same treatment as others who have the same type and stage of cancer. Even so, different people may respond differently, and until recently, doctors did not know why. After decades of research, scientists are now understanding that patients' tumors have genetic changes that cause cancer to grow and spread. The idea of precision medicine is not new, but recent advances in science and technology have helped speed up the pace of this research in modern time. Sir Walter F. Bodmer is paramount figure in the international cancer and genome research field. He has done many pioneering work and been involved with groundbreaking initiatives, including Human Genome Project. Sir Walter Bodmer went to Cambridge for a degree in mathematics, and he did his PhD with Ronald Fisher, a renowned figure in statistics and genetics. After obtaining a PhD in genetics from Cambridge University, he returned to Stanford University in California, where he rose to the position of a professor. In 1970, he returned to UK as the first professor of genetics of Oxford University. He subsequently became the director of research and finally the director general of Imperial Cancer Research Fund in London. Now he is serving as head Cancer and Immunogenetics Lab, University of Oxford. He is a fellow of the Royal Society and was knighted in 1986. Today, we are privileged and taking the opportunity to hear from Sir Walter Bodmer on cancer genetics and precision medicine. I am cordially inviting our keynote speaker today of today's seminar, Sir Walter Bodmer. Sir.
I'm going to talk to you about genetics of cancer and the way we can deal with it and what precision medicine means. And I hope some of you will I think we've got to switch these off. Some of you will forgive me if I cover some fairly elementary basic genetics on the assumption that not all of you are familiar with what DNA does and why it's important. When I started in genetics in the mid-1950s, hardly anyone knew what DNA or genetics was, and now it's mentioned almost every day in the newspapers. Well, I want to start off with some basic facts, a little bit of what I've learned about the contrast between the situation in Bangladesh and what it is in other countries. Oh, I see the computer's there. It still doesn't work. Oh, there it is. Okay, that's my title. So what I show you here is the demographic constitution of the population, the age structure. And I'm comparing it with the United Kingdom, and you'll all be familiar with the fact that Bangladesh is a very young population with something like 50% under 25. Because cancer is mainly a disease of older age, that has a major influence on the distribution of cancers and what will progress later. And it's not generally realized, I think, how important cancer is in developing countries, and not only the developed countries, and how it will become increasingly important. And you can see the very different age structure which gradually is approached as you become hyper-developed as we are in the United Kingdom. And so it's interesting to look at what the influence of that has on the types of cancer that you get. And this is data that, I'm not sure whether these are data that compare the incidence of cancers in Bangladesh with the world average incidence. And there's some striking differences. You have a very high incidence of esophageal cancer relative to elsewhere, lip and oral cancers. Breast and lung are common as they are elsewhere. But one of the commonest cancers, which is one I work on a lot, and which I'm going to use as an example, CRC, it means colorectal cancer, comes very down the list. Cervical cancer, though, is relatively common and is known to be due to the papillomavirus. So there's a challenge here to understand more about the distribution of cancer in more detail as it occurs in Bangladesh, and to try and answer some of the questions of why there are these differences. But I want to start by telling you, well, what actually is a cancer? You've all heard about it. You've nearly all come across in one way or another people who've suffered from cancer, and it's still something we can't deal with in as much as we would like to be able to. And I'm going to talk about colorectal cancer, which is common, and it's a cancer in the bowel, in the large bowel. And the cancer is a lump that grows like this, abnormally, quite different from the normal situation in the bowel. And when the pathologist looks at it, they see this. They take a section of the cancer and they stain it with a dye so that what you're looking at, I don't think this pointer works, does it? What you're looking at there on the left is the normal situation. And you see those regular structures and the dark dye and the little the small things that are illustrated there, they're the cells, individual cells. But when you have a cancer, the whole structure is disrupted, and you see what's down there. Everything is disorganized, and the highly organized growth of the cells is disrupted. And here you can see where you've got the normal situation with those organized crypts, those long structures, alongside the disorganized structures. So that's what the pathologist who does, that's what the pathologist, this way. Okay. Is that seen on the screen? No. It doesn't work on these screens. It doesn't matter. The pathologist sees those differences, and that's the ultimate way at the moment that the cancer is diagnosed. Now, I'm going to start by telling you about an inherited form of cancer. Now, most cancer is not inherited in the sense of being passed on from parent to offspring. It's mostly, in that sense, not a genetic disease. But to introduce the notions of genetics, I'm starting with an example where there is an inherited tendency for cancer. And it's a cancer of the bowel called familial polyposis. These different growths, these are lumps which are precancerous growths, and that's the cancer developing further. And people who have this abnormality have an inherited tendency to get cancer quite early in their 20s and 30s, which is quite unusual for colorectal cancer. And this is an example of the pedigree. This disease was first described at the end of the 19th century, but it was first shown to be clearly inherited in London by a group there in 1925. And this is the pedigree that was shown. And you see a pedigree, these are the symbols for the males, those are the females, and the filled-in symbols are the individuals in the family with cancer. So you can see how it's passed on from the male here to several of the offspring here, and then down here. So that's a characteristic pattern of inheritance. The way that patterns of inheritance were first analyzed by the founder of genetics, Mendel, who studied peas and not humans. And so the question is, how can we understand what's going on in a family like that? And we all start from the fusion of a sperm from the male and an egg. A single cell from your father and a single cell from your mother that brings together the genetic information from the two parents. And the genetic information is carried in the nucleus, the center of the cell, in structures that you can identify as chromosomes. So you see here is these are cells, these are dividing cells, and you can see those lightly stained, they're the chromosomes that you can see when a cell divides. And when you look at them and order them, we have 46 chromosomes, 23 pairs, and then an X and a Y chromosome. So I'm a male, I have an X and a Y, and the females have two X's. And when you put together, marry an X and an XY, half of them are XX, half of them are XY. That's why, roughly speaking, half of the offspring are male and half female. That's the simplest pattern of inheritance. And we know that the genetic constitution is carried by these chromosomes. And it's carried by a material that you've all heard of and has already been mentioned, and that's the DNA. So the main constitution of the chromosomes has as its center the DNA, which provides a code, a chemical code, which when you read it tells you what genes you have in different positions on those chromosomes. And of course, the revolution of the latter part of the 20th century has been the identification of the structure of the DNA, the way it works, and the way we can read that code, and what we can learn from doing so. So that's the basic genetic background that I give you without going into the technical details. So the question is, how can we relate the pattern of inheritance in the family to what happens at the chromosome level and the DNA level? And in the case of polyposis, it was found that there was a single case of an individual with polyposis, with this inherited abnormality, who had an abnormality in the chromosomes. There was a bit of the chromosome missing there. And that's pointed out here. So that suggested that it was in this position where the bit of the chromosome was missing that the genetic information that gave rise to the inherited tendency to get the cancer would be found. And using the DNA-based techniques, you could first of all verify that that was the case. So you could have genetic tests, which are still used but a little primitive, where you can have a piece of the genetics DNA that you know is in that position in the chromosome, and that it goes together with the inherited patterns. So where you see these patterns, that's where you see these individuals. And it's through that sort of technology that you can then eventually read what the code in that position was and identify the gene, which is a sequence, and the protein that it makes, which has the function of the gene, that tells you something about the whole functional nature of why one gets that particular inherited susceptibility. That's the basic way in which a lot of genetics of inherited diseases that you can follow in families can now be worked out, and given the whole sequence of the genome, you can actually read it off when you know where the position is. And the gene that was found here is a gene that turns out to be very important. That's the next picture. the gene that was found here is one that is mutated, that is changed in many cancers, and I'm going to tell you more about that. And just to say something more about inherited cancers, this is another example of a cancer inherited tendency. In this case, you have many different cancers. Colorectal cancers, they're the individuals who have the light blue. So this is the presence of a colorectal cancer. This is the presence of other tumors. These are skin lesions. And this is an inherited tendency, the basis of which was found, the gene was found. So you can see you have mainly colorectal cancer, but also endometrial stomach cancer, ovarian cancer in these families. And in this case, the gene that was abnormal, that was mutated, that led to this inherited tendency, it controls a very important function of the cell. The DNA is replicated and mistakes are made. And we have a very complex machinery that actually repairs DNA. And when that machinery breaks down and you have mutations in the machinery that mean that it doesn't work, so you get more mistakes, then you get a higher tendency to get cancer. And that's why you get this variety of different cancers in that case. Now, I show you this because it's led to the possibility of treatments of cancer in a way that is my first example of precision medicine. So that these are the genes, these are just names. But there are two main genes involved here that when they're defective give rise to this inability to repair the DNA, which leads to the cancer. And it turns out that when you have mistakes in your DNA like that, then the cancer carries a lot of mistaken proteins, a lot of things that can be recognized as foreign within your body. You will know that you have an immune system that recognizes foreignness, and that's part of the major way that you protect against disease, viral diseases and bacterial diseases. And you're also probably familiar with the fact that you can't transplant a piece of skin or a kidney or bone marrow from one individual to another if they're not matched for certain genetic differences. And that's because the immune system recognizes those. So in these cases, the immune system recognizes the foreignness of the cancer and is part of the way that the cancer is attacked by your body as an alien element within the body. But recently, major developments have taken place in ways in which you can enhance the way that the body reacts immunologically to these changes in the cancer. And there are drugs based on what we call antibodies, which can prevent the immune system from being toned down, from being inhibited by the cancer. And so there are now treatments which are actually proving to be very successful for the successful treatment of these particular types of abnormalities. So these are the mismatch repair deficient cancers, and when they're treated in this way, they survive much better. This is a survival curve over a period of months, and the ones that are not mismatch repair deficient, they don't survive so well. But the ones that are enormously helped by that particular treatment. So here you have an example of what precision medicine is. It's using a drug, an antibody, to treat a patient according to their genetic makeup. And that's one striking example of how that can be done. But I want to take you now into the question of, well, what's the next step? The Vice Chancellor, the head of the department mentioned that there are genetic changes in the cancer itself. And I want to take you into that field because that's one of the most important areas of research that tells us what goes on in a cancer. And I want to start by telling you about a very different sort of cancer, leukemia. Well, sorry, before I go to that, I should just make the point that inherited cancers are a very small part of the background to cancer occurring, a few percent. There are genetic tendencies which we study, and that's a topic I won't go into the detail, it's quite more complicated. But the vast majority of cancer doesn't involve an inherited tendency. And so the question is, what happens in these cancers and what can we do? And that's where the genetic changes in the cancer itself become important. So I want to introduce you to leukemia. It was a term that was introduced by a famous German pathologist in the 19th century, Virchow, who was the first to recognize that cancer was a disease of cells in the body when they misbehaved. And leukemia actually means white blood. And if you have a patient with a particular form of leukemia I'm going to talk about, you can see this is a blood collection sample. These are the leukemic cells. They're the white cells of the body. They'd usually be a thin layer at the top. These are the red cells. So this is a huge overproduction of a particular cell type that is the cancer that is causing that disease. And the steps that led to an understanding of exactly what that disease is primarily due to and how it can be treated came again from studying the chromosomes. And this is a picture of the chromosomes at a time when the technology was not quite so good. But it was found out by workers in Philadelphia that there was an abnormal small chromosome that was always present in this form of leukemia. It's called chronic myelogenous leukemia, CML. And then Janet Dowley, who was a distinguished geneticist working in this area, actually after she spent a sabbatical in my lab in Oxford, she actually identified what the nature of this little chromosome was. You can hardly see it here, that this is a little bit different from this. But that's actually the picture from the paper that described this. And what it turned out to be is an abnormality of the chromosomes that was quite distinctive. And here you can see chromosomes defined by these banding patterns. This is the normal chromosome 9. But the chromosome 9 in the leukemia patient had a little bit of extra there, like that. Whereas the chromosome 22 had a bit missing. And what she realized is that actually this was an abnormal chromosome in which there'd been an exchange from here to here that produced that. So that defined actually what was the basis for the key. What's happened there? So this is it schematically. Chromosome 9 is indicated in pink, 22 in blue. If you cut a bit of that off and a bit of that off and rearrange them, you get this chromosome here and that chromosome there. And it turns out that in this region of one of the chromosomes 9, there's a gene that's attached to another gene here. So you're making a new abnormal gene. And it's that gene whose change is driving that cancer. And that's illustrated here. So you're putting together material from chromosome 22 in the filled-in squares with chromosome 9. You form the Philadelphia chromosome. That makes a product that leads to a totally abnormal protein, which is what's driving the cancer. And with that knowledge, a drug was found that inhibits the function of this particular protein. And that drug turned out to be a remarkable way of treating and largely curing that type of leukemia. So here you have actually the fundamental ideas of what lies in a cancer. It's a genetic change in the cancer itself. And that leads to another way of doing precision medicine, of treating the cancer according to its genetic constitution. So here you're finding a drug that is treating a cancer effectively because it's targeting a particular genetic change that is distinctive and is not found in normal cells. So the development of knowledge about cancers has come about through finding what are the mutations, what are the genetic alterations that happen in the cancers that are not necessarily present and usually not present in the germline, inherited from the sperm and the eggs. So we come to say, well, what is precision? What is personalized medicine? And I've given you two examples. One is to try and find out the genetic makeup of an individual and to a treatment of a disease that is due to that genetic makeup and that helps treat the disease from the knowledge of the genetic makeup. And the other, which is distinctive, particularly for cancer, is to find out the genetic constitution of the cancer and to treat the cancer according to its genetic makeup, the genetic makeup of the cancer, not of the individual. And the third example, which I'll come to at the end of my lecture, is where you might have an individual genetic difference in the way you respond to a drug, how you metabolize it, what you do with it. And that's really the third plank of what precision medicine is. If there is a susceptibility that's connected with the way you treat the drug, then that's another way that you can target what's going on. So we have to think of cancer as an evolutionary process of cells within the body. You get mutations that occur, and each mutation gives some advantage to the outgrowth of the cancer. And these happen successively, just like evolution in organisms that leads to, that we know from Darwin's ideas on the evolution by natural selection. But this is with respect to what's happening within the body. So cells acquire mutations at random, maybe increased in frequency by environmental factors. And each mutation that gives the cells an advantage to escape from the constraints of their normal growth and to increase in growth, to grow in metastatic in foreign sites, leads up to a series of what are called driver mutations, the changes in genes that are the most important changes, that drive a cancer to grow more aggressively, to grow larger, and eventually to give the disease that we know as cancer. Now I want to say a little bit more about the nature of the underlying biology in terms of colorectal cancer. The bowel is a tube, and the surface of the tube isn't a smooth surface. It has these things which I've already shown you, sorry. These are individual cells, and these are crypts, these are sort of little depressions in the surface, and all of these individual cells are the cells of the surface of the bowel. And schematically they're illustrated like this. So this is one crypt. And there are cells at the bottom that are the most important. They're the stem cells. They're the cells that divide to produce these other cells, which are still dividing, and these, which are the final cells that do the function in the crypt. And that structure, the whole structure of your bowel, turns over, is replaced by this division process every between 4 and 6 days. And that's maintained throughout your lifetime. But it's these cells here that are the ones that keep dividing. And it's when you have mutations in those cells that things go wrong and you don't get the ordered structure that you've got here, as I showed you in an earlier picture as to what a cancer is about. So we've now been able to study, partly using families, partly using cancers at different stages, so that you can identify a process that goes from the normal surface, goes from an early stage growth, which if you cut it out, is a complete cure. It's not gone anywhere. And each stage is pushed along by another mutation in the gene until eventually when the cancer spreads, and especially if it spreads to other parts of the body, it begins to be more and more difficult to treat. But so long as it's contained at an early stage like this, it can be cured by the surgeon. You just cut it out. If there are no cancer cells anywhere else, you've actually cured the disease. So that illustrates another very important feature. The earlier that you can pick up a cancer, the better you're able to treat it. And that's another area which needs developing based on our understanding of what cancers are. So we have a list of genes. You don't need to worry too much, but we can grow out cell lines. We can grow cells in culture from cancers. And in my lab, we have a collection of 100 or so of those cells. And this is the proportion of the different cell lines, different cancers that have this particular gene mutated, a very common one, P53. APC is the gene that underlies the inherited tendency that I talked to you about with all those polyps. And that's mutated in something like 80% of all cancers. This is another one. And then there are other genes that sometimes mutated, but at lower frequency. But it's these ones, they're the drivers, they're the ones we've got to try and find out how to deal with it. And there's an interesting feature about this. The cancers that come from those situations where you have an inability to repair, the mismatch repair as we call it, you get a different distribution. So you get some genes that are very highly mutated and presumably selected for in cancers that have this repair deficiency, which you don't find in cancers that aren't. And these are the cancers that I mentioned can be treated by these antibodies that are called checkpoint inhibitors, which is a major advance in the way of treating cancers that got the Nobel Prize 2 or 3 years ago. So I want to give you some more examples of how you can direct treatments that take into account the constitution of a cancer. And nowadays, the most common drugs, the most widely used drugs for the treatment of cancer have become these antibodies that the body produces, that an animal can produce, that recognize very specific genetic changes that lead to changes in the protein. And one of the factors that keep cells going, there are growth factors. There are things that attach to the surface of a cell that stimulate it to grow. And if you interfere with that process, you interfere with the growth, and that's a way of treating it. So one of the treatments for a colorectal cancer is to give an antibody that interferes with that growth process. And each of these is a different cell line. And here is measured the effect of this antibody on the growth. So there are a lot of tumors that don't respond very well. And there are a few that respond very well. And you can see that the ones that respond very well, they have those blue rectangles above. They're the ones that do not have mutations in certain of the genes that are listed there that are commonly present otherwise. So you can only make this treatment effective if you give people the drug who don't have mutations in those genes. So that's another example of precision medicine, of giving the treatment according to the genetic makeup of the individual. It also turns out that this mismatch repair defect is present in the germline, is present in the cancers, many cancers, without necessarily having been inherited. And recently, it was realized that prostate cancer, which if you go old like I am, you've got a much higher chance of having a prostate cancer. So you don't find much yet in Bangladesh because of the age distribution. But it eventually becomes the commonest cancer almost in males, apart from lung cancer if you smoke. And a small proportion of these have this repair defect. And that's another example of where, if they have that, you can treat them with these antibodies that stimulate the immune system to deal with that cancer. Now, I haven't talked yet about how you can deal with treatments that are related to an inherited tendency of how you respond to a drug. Abacavir is a drug that's used for the treatment of people who have HIV infection and AIDS. And it's a very successful component of the way that AIDS is now treated. But it turns out that there's a small proportion of people who have a particular genetic makeup of the genetic variations that are involved in the immune response system, that are B57 as it's called, that have this is an identifiable genetic makeup of an individual. And if you have that and you get that drug, you have very severe allergic responses, which in some cases can be lethal. So you shouldn't give that drug to an individual who has that makeup. So that's another example of precision medicine at the level of your genetic makeup as you've inherited it, where you're guided by your genetic makeup in what drug you should take. Another example, not in the cancer field, is a widely used drug if you have gout, which is a very unpleasant disease which I suffered from for a little at one time. And there's a drug called allopurinol. And if you have B58 rather than B57, you get a very severe allergic reaction. And many years ago, I diagnosed myself as having that, and you must avoid giving allopurinol to anyone who has that genetic makeup. So that's an example of precision medicine in another case involving allergic reactions, inappropriate reactions to drugs. So I've tried to give you here then examples of the way that you can deal with precision medicine in the case situation of a cancer. According to whether it's inherited, you can do something about it. And there are many inherited diseases, of course, other than cancer, where people are beginning to find, even if it's quite a rare disease, ways in which the disease can be treated from a knowledge of what the gene is that's gone wrong that's given rise to the inherited disease. Cancer is almost unique in the fact that it involves genetic changes in your somatic cells, in the cells in the body that have nothing to do with the sperm or the egg, and where you get this genetic change, evolutionary process that leads to the disease of cancer. And fundamental understanding of a cancer will only come from studying what are the genetic changes and what are their consequences, and how can we use that information in the context of treatment to target a treatment according to a cancer's particular genetic makeup. And then lastly, this example, of which there are many more examples now in other areas, where your genetic makeup influences the way a drug behaves. And so that can either tell you, well, you're someone who will respond properly to that drug, or to someone for whom that drug has a severe adverse reaction. Well, there's large numbers of studies, of course, now that have been and are being done in order to understand more about the genetic makeup of cancer. And I just finished with this slide, which is one program that was developed in the UK, which has been somewhat at the forefront of developing large-scale studies of genetic changes in individuals and in cancer. So this was a program of doing whole genome sequencing on 100,000 individuals, of whom a quarter or more were cancer patients, and which has given rise to a catalogue of genetic changes that you can find in different sorts of cancers as they occur in the United Kingdom population. And recently in the journal Nature, there was an issue that had several papers where people have got together on a worldwide scale and looked at the genetic changes that happen in a whole range of cancers in different countries, and giving a picture of what are the key changes, the key driver changes that are important for different types of cancer. And this is certainly a thing that I think could be done and needs to be done more in this country, to understand more, as I started at the beginning, about the nature of the cancers that you have at higher frequency at the moment. What are the genetic changes and what are the factors behind them? Certainly most of the lung cancer will be due to still smoking. That's the one major preventative measure that one can take. But those that do occur, you will understand by the nature of the genetic changes, and that's the way we'll learn how to treat them better. So I think that's where I'd like to finish, and hope that you've got something of a flavor, and if you all understood it before, you'll have to forgive me.
Thank you, sir, for sharing this immense knowledge with us. Now we will proceed to question and answer session. Audience are welcome to put forward their question. Please welcome.
We know that you have tell us many things about cancer genetics and precision medicine, but I want to go some different aspects. Like we know that caspase enzyme, that is cysteine-dependent aspartate-directed protease, that have the role in programmed cell death. And due to deficiency of the caspase enzymes, cancer may happen. So in the present situation, what are the latest development in cancer, in caspase targeted cancer treatment? Can you, caspase targeted cancer treatment, what is the latest situation in the world?
Caspase, for those of you who don't know, apoptosis is a form of what's called programmed death. And in the normal process of differentiation, cells have to die after they've differentiated eventually. And it's probably the case that decreasing apoptosis, decreasing the rate of programmed death in cancers, is one of the mechanisms that leads to cancer more. And it's thought, for example, that P53, which is essentially the commonest mutated gene in cancers, really works by reducing the rate of apoptosis. But the thing to target is the P53, not so much the apoptosis. So I'm not aware of any successful drugs that treat cancer by treating the apoptosis directly. I think we have to start a little bit upstream and treat the genes that lead to the changes in the apoptosis.
What's the relation of the PSA with the prostate cancer?
And is a marker for the presence of a prostate cancer. I don't think anybody really knows the significance of its function, but there's also a prostate-specific membrane antigen, which is expressed on the surface. So the way to attack prostate cancers, one possible way, is to attack the membrane prostate cancer, because that can be attacked by antibodies that will kill the cells. But I don't think attacking the function of PSA, I think it's one of the kallikrein type, as I remember, proteases. I don't think the function is other than indicating that this is a cancer of a prostate rather than a cancer of the bowel or the breast.
So during localized proliferation, that means localized cancer, before metastasis, we have some different treatment pattern. But after metastasis, it has no any neovascularization, no any new blood vessel formation. So after metastasis, a floating cell, how easily can adapt to the normal cell and to get nutrient and any other oxygen supply, and then again it can proliferate more rapidly than the normal cell.
Issue of metastasis, which is very important. As I've emphasized, when a cancer is localized, essentially you can treat it by targeted radiotherapy or by surgery. One of the problems with breast cancer is even when you do that treatment locally and you think it's still localized, there are still deposits of cancer cells in different parts of the body that can grow even after 10 or 12 years. But the question you're raising is how can you target the metastasized cell if it's different in the pattern of growth from the normal cell. And of course it's because it's spread that it's difficult. But the metastasized cell still carries the driver mutations of the primary cancer. So I think actually targeting metastasis specifically may not be an easy thing to do. And it's well known that a metastasis will grow because a cancer cell is growing in an alien environment that's not usual. And it induces local changes. In the case of prostate cancer, colorectal cancer, the myofibroblasts are activated and help the cancer to grow. So there may be ways of targeting that interaction to help deal with the cancer. But I think still attacking the primary driver mutations that are present in the original cancer is still the best way to go, even for treating a metastasis. But it's got to be done with a drug that will reach all the different areas where the metastasis is taking place. So you can't do a localized treatment unless you've got localized lesions, say, in the brain or the liver, which you can try and treat by targeted radiotherapy or even targeted surgery. But there's a lot of work on metastasis, a lot of claims of what are the key features, and I think it's still not completely understood. And I think often too much emphasis is given on the ability of a cell to get to the foreign site rather than its ability to grow there.
Sir, a young lady with leukemia is surviving for the last 2 years without any abnormal symptoms, silent symptoms. And what could be the precision medicine, and how how long she can survive without any problems.
In the end, the precision medicine is going to be by knowing for each patient
Yes.
what the genetic changes in that patient are. So, for instance, I've been involved in development of antibody drugs in colorectal cancer in collaboration with the company Roche. And there's an antibody there that attacks a particular cell surface component that's present in about 20% of different cases of colorectal cancer. So that will only work if you actually have that particular expression. And so that's the way the treatment's going to go, by finding out what's present there. But a lot of the current treatments are not targeted. And the checkpoint inhibitors are targeted, because there you know, if you've got cancers where you've got lots of mutations, like in melanoma because of sun, or lung cancer because of tobacco smoke, or the mismatch repair defect, then that itself is an indicator that the checkpoint inhibitors will work, but they still only work in a proportion of the cancers. So I think the future has to lie in understanding more targeted treatments. In my view, that a lot of that can come from targeting according to what's on the surface of the cancer or what's inside in rather particular ways. And it may be that what you target is not something that's driving the cancer, but happens to be present more in the cancer, and therefore it's a handle by which you can kill it.
In cancer treatment, there are some cytotoxic anti-cancer medicine, like cisplatin, oxaliplatin, doxorubicin. So nowadays, some monoclonal antibody have been launched worldwide. In future, will this monoclonal antibody, which works at target specifically, so will monoclonal antibody will replace the conventional cytotoxic drug? Because this cisplatin, oxaliplatin has some side effects.
Is they just kill the patient, the cancer, a little more than they kill the patient. It's a very delicate balance, because they're drugs that are generally toxic, but for one reason or another, the cancer is more susceptible to them. The aim of the antibody treatments is to have things that are much more specific to a feature of the cancer cell itself. And actually, the talk I'm giving tomorrow to a group of biotechnologists organized by Mushtaq is what I see as the developments in the way that monoclonal antibody therapies can develop. And I think they will play an increasingly important role. The other area, many of you may have heard of CAR T. CAR T is a way of making your T cells, which recognize viral things on the surface and so on, making them recognize cancers. But so far that's only worked for the blood cancers because of the accessibility that they have to the CAR T cells. So I think that a major direction for the future will be more use of monoclonal antibodies. And already there, Avastin, there are a whole series of Herceptin monoclonal antibodies that have been being used quite widely already in cancer treatment.
Okay, there are some oral anti-cancer product now, which are called tyrosine kinase inhibitor, gemitinib, imatinib, that is the tablet form. So most of the anti-cancer drugs are injectable forms, but nowadays anti-cancer drug in tablet form is also available.
Yes,
so
well,
what about the tyrosine kinase inhibitor?
Is an inhibitor of a tyrosine kinase activity of an oncogene able. So there are a number of those drugs, but I wouldn't call them cytotoxic drugs. They're targeted drugs to particular enzyme functions, and they have been quite successful. But the number of such opportunities is still somewhat limited. But there's certainly, I mean, if you know about it, there are drugs that treat people with BRAF mutations, for example. And those are important. But I don't classify those with cytotoxic drugs. I think the cytotoxic cisplatin and things like that, they're the ones that are overall toxic, but the cancer is somewhat more susceptible than others. And it may be that they can be used in combination with antibiotics. No single treatment of a cancer will ever work. Cancers will always develop resistance, so you must have multiple treatments to lower the probability that you'll get a resistance to all the drugs you use.
Country like us, Bangladesh, we don't have that kind of resource, and it's very difficult for us to take that kind of grand project. So what is the future for precision medicine for a country like Bangladesh? What can we do?
Even though you're not as developed a country as the UK, I don't think you should ignore the possibility of doing some of these large-scale studies. They can be extremely informative, and although the cost seems large, relative to the cost of cancer care and other medical care, it's not so large. But in terms of the immediate possibilities, there's so much known about genetic changes that you can do a lot of targeted sequencing relatively cheaply and with newer technology as we've been discussing. And I think that's the way to go at the moment, is to find out more about the genetic changes in the cancers that are unusually common in this country. And probably the changes that are common, like in lung cancer, or as you get an increase in colorectal cancer, will be the ones we already know about.
Hello, sir. I am Kona,
Yeah.
student of pharmacy department. My question is, you have shown that many types of cancer is occurring according to uneven cell development in many organs. Why the ratio of getting cancer is high only in breast and lung?
In the lung, the reason that there's a lot of lung cancer is mostly because of smoking. And when smoking is reduced, as it has been remarkably in the UK, the incidence of lung cancer goes down. But there's still, even amongst where smoking is the largest cause, there's still a 10 or 15% of lung cancers that are not due to smoking. But we don't know why cancers are commoner in some organs than others. For example, the large bowel has a high rate of cancer, but the small bowel, which turns over just the same way, doesn't. We don't really know why that is. It's an observation. We can try and find out why. Maybe that tells us, maybe it's something to do with the microbiome, I don't know. But I don't think we know why cancers in some cases are more common than others. I mean, cervical cancer, we do know a lot about that. It's a sexually transmitted virus that causes that. Breast cancer, you can say you'd expect it to be more common in females than males. It does occur in male cancers, but much less frequently. So it's a cancer that's due to the function of the breast, and it's related to some extent as to the history of a woman with respect to how many pregnancies she's had and whether she's breastfed and things like that. So in some cases we have an idea. Stomach cancer is almost always due to Helicobacter pylori, and which is a major cause of ulcers, of course. And as hygienic conditions change and you get rid of the presence of Helicobacter, ulcers go down and stomach cancers become relatively rare in developed countries like the UK, where previously it was common. So in some cases you know, but in other cases, you know, a large element of cancer is chance. Mutations occur at random. You can increase their frequency, but they don't occur according to the function. So in some cases we know, in some cases we just don't know. And for instance, if you take the equivalent of polyposis in the mouse, there's the same mutation. It causes cancers in the small bowel, not the large bowel.
It has been an absolute honor for me to be present here in your talk. So I'm Dr. Muhib from Bangabandhu Sheikh Mujib Medical University. I'm an oncology resident. So I'm asking quite a bit clinical question, that is, do you really routinely see mutation of microsatellite instability in case of colorectal cancer in early stage or before treatment of the colorectal cancer?
Occur before metastasis. Yeah. Most of these mutations are driving the early stages. And by the time you get to metastasis, cancers grow so rapidly that there's not a lot more genetic change. So mismatch repair mutations occur in about 10 to 15% of sporadic, non-inherited colorectal cancers. And again, I think we don't know that that's driven by anything. It's just the chance that that's the way the mutations happen. And so they occur early enough, because as I say, all the changes we're looking at really occur before you have metastasis. The major changes that are found occur before metastasis. So in that sense, they're on the early side. And of course, that's why to some extent you can screen in Lynch families the inherited mismatch repair defects. You can do some screening of looking for polyps, which is what they do for polyposis, which is one way of reducing the risk. I mean, that's if you were a targeted treatment. If you know in a family that there's an individual who has the polyposis mutation, then you screen them regularly, you remove the polyps, you remove the risk of them getting the colorectal cancer. And to some extent, that can also be done with mismatch repair, but the number of polyps you produce isn't as large. I don't know whether that answers your question.
Yeah. So the reason why I asked that question is that due to limited resources in our country, we usually test for mutation for MSI in case of metastatic patient, metastatic colorectal cancer, as because you know that in case of metastatic colorectal cancer, 5-FU and leucovorin, the first line treatment are not work that good. So another thing is that, sir, I must thank you for your precise and simplified presentation, and I must invite you to my department, the Department of Oncology, if you have time. Thank you, sir.
Sir, there is a many gene mutation which cause specific kinds of cancer, and scientists are trying to recover this problem through different genetic recombinant technology, CRISPR gene editing, or modified gene insertion, suppose through AAV vector or different kinds of vector in preclinical trials. That means use mice, rats. So, what is the future prospect for clinical trial? If we directly insert any modified gene into the body locally, is it possible to recover any kinds of gene?
I don't think genetic engineering of that sort is easily going to play a role in cancer. Because if you want to, say, correct the P53 mutation, you've got to correct it in all the cells. And the chance of really doing that with externally administered treatments is not very high. So if you say were to insert DNA sequence into a virus like an adenovirus that attacks lots of cells, I think the chance of hitting every cell is not very large. So I don't think that's the way to go. One of the particular advantages of immune therapies is that very often when they target the cell that carries the thing you're targeting, they actually stimulate the immune cells to attack cells in the cancer that don't have that target. So you spread the effect, you get a bystander effect. And I think that's very important, because if you only target a subset of the cancer cell, or you don't target the stem cells, the cancer will just regrow.
Thank you so much, sir, for your nice presentation. And actually, sir, I was in hesitation whether to ask this question or not. And I was waiting to get answer from another question. Actually, my question is, I will tell in brief. Cancer genetics and precision medicine is about to identify individual personal genetics and treating their genetics, individual genetics, to remove the cancer cell. But don't you think this is precision medicine is just only for rich people, not just for poor people? Because identifying individual genetics and treating himself by his genetics order, isn't it very costly? And what are the future initiatives to make it cost effective?
First of all, the most expensive thing in a cancer is treating a patient for a long time who eventually dies. And that's expensive. So if you prevent the need to do that, then you save quite a lot of money. Obviously, prevention works better. Secondly, it is not expensive to do these genetic tests. There are ways now of doing them in a way that you could certainly afford here. And I think you've got a need to think about what developments can make those sorts of tests more readily available at manageable costs. Thirdly, people think of antibodies as expensive. Antibodies are now produced by the ton. They are produced just as cheaply as many other medicines. The companies are charging large amounts for them because they're trying to recover their costs of developing them. But eventually those costs have to come down. And I think if you're really going to be successful in treatments, then you've got to be prepared to spend some of the money that's involved in those treatments, having done everything you can for prevention and early detection in the first place. So that would be my answer. I think it's an inevitable need to develop them in a way that can be afforded, and I'm sure that Bangladesh will go in that direction, that it'll be able to do that.
Now, I am cordially inviting our special guest of the session, Professor Dr. S.M. Mahbubul Haq Mujumdar, to take a few words among the audience. Please welcome, sir.
We feel proud to welcome Sir Bodmer, who is a renowned scientist in the field of cancer and immunogenetics of the whole world. We know that nowadays the hand of cancer is spreading all over the world very quickly. And we observe and we listen many important things about the cancer and genetics from Sir Boulder. And from this speech, and our scholars, researchers, and students will be highly benefited, and they will interact with each other about the research activities on this field. And we are very much delighted and thankful to the keynote speaker for his gracious presence despite his busy schedule. Thanks to the Department of Pharmacy and Public Health for arranging such a meaningful seminar by your active participation. Many, many thanks to the members of each and every committee for organizing this seminar. So again, my warm thanks to Sir Bodmer for coming to Bangladesh and delivering a very, very meaningful and very attractive speech. So thank you very much. Thank you.
Thank you, sir. It's time to show gratitude to our honorable keynote speaker. We would like to request Chair of the session, Professor Dr. Yusuf Mahabubul Islam, Honorable Vice Chancellor, Daffodil International University, to hand over the crest and gift to our honorable keynote.
There's a famous saying that you know something only when you can explain it to your grandmother. Yes. So thank you, Sir Walter, for putting the thing so simply for us today. So let's give him a big clap again. And there was a few things about limitation in our resources. But I would like to say that when you want to do something, you can get it done. It's not the resources. If we keep looking at our resources, we will never get anything done. So one of the things pointed out by Professor Walter was the differences in the cancers in Bangladesh with the rest of the world, in particular UK. And you will notice that the cancers are of the esophagus, yes, and lower abdomen, yes. The majority of cancers in Bangladesh are from this area. So there is scope for, as he pointed out, that when dietary habits change, this can change, as it has in other countries. So there is scope to find out the relationship between our dietary habits, our food impurities that we introduce in Bangladesh, and these cancers. And if we do that, maybe we will find out why we are so different from the other countries of the world. And this precision medicine, because one of the things pointed out was the major cancers are not hereditary. They're developed through our own habits in our own lifetimes. And that is why individual medicine, individual targeting through precision medicine is important. So one of the role of personal hygiene, personal food habits, and the food available in our country, it is very important. As you know, one of the impurities is formalin. And one baby, the body did not rot even after many, many days because the body was full of formalin. So formalin. So what I gathered from the speech, and I hope you gathered, is that there is a lot of scope of research in that area, and we can always connect with Sir to find out ways to do collaborative research in this area. Is that yes? Yes? And you see that he has spent his lifetime, Professor has spent his lifetime doing these things. And we thank him for a second thanks for keeping at this target area of reducing cancers among society. Thank you, sir.
It was really a meaningful session. Thank you all for coming and making today's session successful. We are really thankful to you, especially those who came from different organizations, especially pharmaceutical companies and academia. Thank you again.